Andreas Moor received his MD degree from University of Bern in 2010. Then he got his PhD degree from EPFL in 2014. And after that, he did postdoc in the Weizmann Institute of Science in the laboratory of Shalev Itzkovitz on spatial science or biology using cool technologies. And then in 2019, he started his fellow-like position, which they call in Europe SMSF professor position in the University of Zurich. And then in 2020 June, he started his own research team as a tenure track professor in the systems physiology department at the ETH Zurich. And currently, he's leading a team doing spatial biology projects, and he will be talking about the technologies and the cool science behind it today. And we are excited to hear about his work in his lab. And without further ado, I think I'd like to turn to Andreas.
Thank you very much for the kind introduction. Can you see my screen?
Yes.
Excellent. Good. So I'd like to thank, first of all, Ahmet and Rong for the kind invitation to talk here today. And I think I have been following the first and the second series of these talks, and I've learned a lot because this field is very much in motion. There's a lot of exciting new stuff happening. So very excited to be here. So my lab is interested in how single cells collaborate within tissues and how they can then jointly achieve their common functionalities. So as you heard, I just recently started my lab at ETH. Most of what I will talk about here today is actually work that I did as a postdoc in Shalev Itzkovitz's lab at the Weizmann Institute. And I saw that a lot of the talks and topics of these third series will focus on spatial methods. So I will actually focus a little bit more on the spatial tissue biology today during my talk. Now, I will mostly talk about the small intestine. I think it's really fascinating organ. And here you see this scheme where we have a highly structured epithelium that consists of those finger-like protrusions that massively enhance the surface area that is available for absorption. Here you see now a cross-section scheme. And this tissue has a really beautiful spatial organization. You have these crypts, which are deep protected niches for the intestinal stem cells. They sit at the bottom. And this is actually the most active adult stem cell compartment that we have. So they divide about once a day, and their progeny is then slowly pushed upwards. And as it migrates upwards, it differentiates and acquires functions. And after 3 to 5 days of migration, it reaches the tip of the villus where it's then being shed. This tissue has to fulfill many, many different tasks, and my research studies how these tasks can all be fulfilled in an efficient manner. So for the first part of the talk, we will look into a cell, into the intracellular realm, and look how different functions take place in the basal versus the apical subcellular compartment. For the second part, I will then zoom out and look at division of labor across different cells in the intercellular realm. So let me start with the first part about intracellular RNA localization. Here you see an image of a migrating fibroblast. This cell moves from the left to the right of your screen. And in red, you see here beta-actin mRNA, and in green, we see beta-actin protein. And you can see that this RNA is not, evenly spread across the cell, but rather enriched here at the leading edge of the cell. And that's exactly where we need new beta-actin protein for the assembly of the filaments. So the cell uses this localization of beta-actin mRNA to the leading edge so that it can locally translate beta-actin proteins exactly where that's necessary. So that's one way of how cells can use localization of RNA. Another example is what you see here, where we have a very polar cell type, a neuron, and neurons can be very long. So if you have here down in this axon a signal that gets into the cell and you want to respond, you could just send the signal all the way back up to the soma, induce transcription, translation of the response product, and then ship the response product all the way down again. This takes a lot of time. So alternatively, what the cells actually do is they localize the RNA to the distal part of the cell, and they can then very quickly locally translate that into response proteins, and thereby enable quick and efficient responses to extrinsic stimuli. So this is not just a few RNA molecules that are having these bizarre localization patterns. A really nice study from about 13 years ago looked at Drosophila embryos and showed that a large fraction of the transcriptome is actually localized in these single cell stages of the embryo. So we in the lab are using a lot of single molecule RNA fish. This is a technique that most of you have heard of, where you use little fluorescently labeled oligos that are hybridized onto RNA molecules. This is not amplified, it's a linear amplification, so you basically see single RNA molecules as these diffraction delimited dots, which means you can actually count single RNA molecules, and it gives you a very good information, a very quantitative information of gene expression in space. So we were using this technique and looked at specific transporters in the small intestine. Here you're now looking at the villus. This is this part that I showed you before. And we looked at GLUT2 mRNA. This is an important transporter of glucose. And we saw that these RNA molecules, so here is one cell, these RNA molecules are not evenly distributed, but for this RNA, they're actually enriched on the apical cell side compared to the basal cell side. So we were wondering if this is somehow related to the efficient function and quick responses to extrinsic stimuli in this tissue. And we wondered what the extent of RNA localization is in this tissue and how it's being controlled and what its functions are. So SM-FISH is really nice for looking at 2, 3, 4 RNA molecules at a time, but it definitely requires that you choose a few genes. You can't get a transcriptome-wide overview. So to do that, we then worked with laser capture dissection and RNA sequencing. So laser capture microdissection is a technique where you take a unfixed tissue piece, create a cryosection, and then you use laser power to draw and cut specific areas of interest. So I was isolating apical cell fragments and basal cell fragments, and then you can catapult them out of the tissue, and you can use low input RNA sequencing technologies such as, for example, SMART-seq2, which are adapted for single cell RNA sequencing, but they can also deal with the quantities of what we get from these laser capture segments. The challenge, of course, is that the RNA is a bit more degraded. So this is non-trivial to get good optical quality for cutting if you go into the subcellular space, while still preserving enough RNA for transcriptome profiling downstream. But after some optimization, we managed, and we got the following result. So here I show you the transcriptome that we recovered. On the y-axis, you see the apical bias. So the higher up here, each dot is a gene. The higher up here, the more polarized to the apical side. And here you see the expression level. And we saw in red that about 1/3 of the highly expressed transcriptome is non-randomly localized and either enriched on the apical cell side or the basal cell side. And then we went back and we designed RNA FISH probes that you can see here. So we picked 3 apical genes, designed FISH probes, and validated that they were really enriched on the apical side. And then we took 3 basal genes and validated the same thing. This is a qualitative look. We then did some co-stainings to show that within the same cells, so here you have, for example, in one enterocyte, within one enterocyte you have apical transcripts and basal transcripts that are being co-expressed. And we then also quantitated many different transcripts that have different polarization patterns, and we showed a very nice correlation between the RNA sequencing apical bias with the single molecule fish apical basal bias. So two completely independent techniques, and that tells us that we can actually trust this. This we did for dozens of genes, but not for all of them, but we can therefore trust the RNA sequencing data from these subcellular patches. So all of these experiments were done in mice, in tissue sections from the gut. This, of course, is a pretty tough model to work with for mechanistic studies. So the nice thing about the small intestine is that we know quite a bit about the stem cell population. And research from the lab of Hans Clevers in Utrecht has identified a culture protocol where you can take the stem cell niche, put it into a 3D gel-like matrix, into a matrigel, and then you can cultivate these little mini guts that have crypt domains that correlate to the stem cell niche of the tissue and little in-between areas where the more differentiated cells sit. And this model is polar because we need an apical basal polarity. If you go to a normal 2D cell culture model, you're not going to have that to assess intracellular RNA localization. So we established then single molecule fish conditions in this tissue, and actually just lately published also a methods paper. So if you're interested in doing single molecule fish in whole mount or cryosectioned organoids, have a look at this method paper. And we showed that what is basal and apical in the tissue is also apical or basal within the organoids. So that's a good intracellular RNA localization model for the small intestine because it mimics the localization patterns that we previously saw in the mouse. But of course, we now can manipulate. It's primary culture. It's not as easy in normal cell culture, but still, we have that in vitro. We can use inhibitors. So here, again, in more detail, an RNA that is basal in vivo is also basal in the organoid. An RNA that is apical in vivo is also apical in the organoid. Now, when we have an intact cytoskeleton, these RNAs are there, and we speculate based on what is known about RNA localization, that these localization patterns are mediated by the cytoskeleton, and here we namely checked the microtubule network. So these are vehicle-treated organoids where we see the normal patterns. When we then use nocatosol, which depolymerizes the microtubule network, we get complete loss of these polarization patterns, telling us that either the maintenance or the active transport is mediated by the microtubule network. Now, the next question was, well, RNA is going to be translated into proteins. So if these RNA molecules are localized to specific areas within the cell, where is the translation machinery sitting? And as a proxy for the ribosomes, we then designed the SM-FISH probe against ribosomal RNA. And as we show here, this is actually enriched on the apical cell side. So the ribosomes are about twofold enriched apical versus basal. The mitochondria, on the other hand, are enriched on the basal cell side. And then we used a technique to measure translational efficiency in a genome-wide manner, which is ribosome footprint profiling from Jonathan Weissman's lab that some of you might be familiar with. This is the technique where you basically isolate all of the RNA that is covered by ribosomes, and you can then, for each transcript, calculate the ratio between how much of it was covered by ribosomes and probably translated to how much of it was there. And you get this translational efficiency score. And here I plot this translational efficiency score for all of our transcripts on the y-axis. And on the x-axis, we have the localization. So we know that the ribosomes themselves are enriched in the apical side, and transcripts that are apical are translated more efficiently than transcripts on the basal side. That correlates nicely. Now, this is all a static situation. We were then wondering, well, what's happening when we dynamically change the metabolic state? So we took mice and fasted them for 5 hours and collected the gut for localization and translational profiling. And in parallel, we took mice where we refed for 2 hours. And I'm not going to go into the results in depth, but we saw that one group of proteins, or basically RNAs that encode ribosomal proteins, are shifting from being biased to more basal in the fasting situation to more apical or more balanced to apical in a refeeding situation. And that also correlated with an increase of their translational efficiency. And when we wait a bit longer after refeeding, we see that absolute protein synthesis is going up. So our model was that, we have these stored basal transcripts that encode ribosomal proteins in the fasting state. And we have this translational hot zone in the apical side. And upon refeeding, they shift their polarization pattern, and that correlates with an increased productions of ribosomes and increased production of absolute protein synthesis. Now, we thought initially that this is quite far-fetched because the newly assembled ribosomes would again have to be assembled in the nucleus. So we were very excited to see a paper that provides evidence of local translation of ribosomes in the axon and then direct incorporation of the newly synthesized ribosomal proteins into ribosomes without going into the nucleus. So we're definitely following that up as some future projects. So in that first part, I showed that in these cells, a large part of the transcriptome is non-randomly localized in a spatially controlled manner, and that microtubules are involved in these localization patterns, and that the translational machinery itself is enriched in the apical cell side. And in a dynamic way, this might enable rapid responses to changing luminal environments. Good, now let's shift gears for the second part of this talk. Now we're going to zoom out from the intracellular realm and look at intercellular division of labor. I told you already that this tissue has many, many different tasks. The enterocytes have to absorb carbohydrates, fats, peptides. They have to keep the microbiome at bay. They have to secrete hormones and so on. So in the second part, our question was, is this tissue doing all of these functions in parallel in the same cells? So is each enterocyte a generalist? Or do we have subspecialization within the enterocytes, that some subpopulations do different tasks? Now, this is ultimately a single cell problem. So you could just take the tissue and dissociate into single cells, perform single cell RNA sequencing, and then you see all of the molecular states that these cells are in. But I told you before that this tissue has a beautiful spatial organization. We know where the stem cells sit, and since the cells are migrating upwards, their position along the villus correlates with their age. So if you dissociate the tissue, you lose all of that spatial information. So therefore, we were looking for methods that can, prevent this loss of spatial information. Now, the method that we were drawn to is spatial reconstruction of single cell data. So the concept is as follows: you take a tissue, you separate it into single cells, and in parallel, you take pre-existing knowledge about landmark genes. So landmark genes are genes that you, for example, here in red are expressed at different places within the tissue or the organism of interest. So this red gene is at the head, and this blue gene in the limbs, and this green gene at the rear of the embryo. And then when you do single cell RNA sequencing, you look into each cell and its expression of these landmark genes, and you can therefore reconstruct where that cell must have come from. The red cell must have come from this area of the embryo and so on. So this process of reconstructing single-cell RNA-seq data was pioneered by two back-to-back papers in 2015. So one was the labs of Alex Scheer and Aviv Regev, Rahul Saticha, that were looking at zebrafish embryos. And the other one was the lab of John Marioni that was looking at ringworms. Now they used gene expression patterns that were derived with classical fish, which is a kind of a binary technique for something is expressed yes or no. So we reasoned, going back to the same slide that I showed before, that by using single molecule fish, which is a much more quantitative method because you can really count single RNA molecules in the tissue, that we could be much more, we would have much more dynamic range of RNA expression in a spatial manner. So the proof of concept where we used single molecule fish RNA gradients to reconstruct single cell data was actually in the liver. So you can have a look at it. I'm not going to go into details. I just want to show you the principle. So we have this one-dimensional axis in the tissue between the central vein and the portal node. And along this one-dimensional axis, several different genes have different profiles, different expression gradients. And we took pre-existing knowledge about these gradients and then quantified them by single molecule fish so that we got the map, an expression map here for these 6 marker genes along this spatial dimension of interest from the central vein to the portal node. And this enables us then to query these genes in the single cell RNA expression space and then place the cell along this axis, and we can then get the spatial gene expression atlas along the axis. So that's the concept. And now I wanted to do that in the villus epithelium, but there the problem was, well, we didn't know any of these landmark genes. We didn't have prior knowledge about how the tissue looks like in terms of gene expression. And of course, nowadays one could use Visium or other techniques. Back then, we actually used laser capture to identify the bulk RNA-seq landmark genes to reconstruct. So how does that look like? Again, same protocol as before. I take unfixed tissue, I stain it, and then I segment the villus into 5 equal compartments, isolate them with laser capture microdissection, and then perform bulk RNA sequencing. And then I pick the genes that have either a pattern that they're highly expressed at the bottom or highly expressed at the top of the villus. And these are now unsupervised genes with a gradient that we can use to calculate for each cell and reconstruct where it must have come from. The algorithm that we used was very simple. We just summed up the expression of the top genes and divided that by the expression of the top plus the bottom genes. So we got this arbitrary spatial coordinate between 0 and 1. And then we actually used the bulk sequencing data from these 5 different segments that we got by laser capture microdissection to then use these as borders. So we artificially binned the data into 6 different bins. Now, when you do laser capture microdissection, you get noisy data of a few hundred genes because you're dealing with unfixed tissue. You're using lasers to isolate. So you're degrading the RNA, and you can only really discover the tip of the iceberg of the genes that are spatially regulated. But when you then move to single cell data and you reconstruct, now you get the full transcriptome. So here you see 8,000, more than 8,000 expressed genes. Each row is a gene. And on the x-axis, we have the different bins. So the crypt, bottom, and to the tip of the villus. And if you look at this figure, you see immediately, now this is all one cell type. We do all of this in enterocytes. This is not a mixture of different cell types. So the spatial information really determines the expression level in a significant manner of 80% of all genes. There's barely any gene that is constantly expressed in this one cell type from the bottom of the crypt to the tip of the villus. So if you were to think of qPCR times, it would be really tough to pick a proper housekeeping gene because 80% of them are actually changing as we simply migrate along the villus and migrate upwards. So that all happens in this time of 3 to 5 days. Now it's all a computational prediction pretty much based on this reconstruction. So we picked a few genes of interest. At the top you see ADA, adenosine deaminase, which is important in purine metabolism. Here you see a gene, SLUC2A2, so the glucose transporter. And here you see REG1, an important antibacterial peptide. And so this gene goes up at the very tip of the villus. This gene is expressed mostly in the middle, and this gene is high at the bottom of the villus. And then again, we designed SM fish probes and we validated their expression patterns in the intact tissue, and you see the result here. So ADA in blue is really at the tip, SLUC2A2 in the middle, and REG1 at the bottom of the villus. We did this for many genes, and again, you see here a pretty nice correlation, always blue, the single-cell RNA-seq reconstruction, and on the bottom you have again, the x-axis is always from crypt to villus tip, and in red you see the completely independent data of image analysis of SM fish, and you see a nice correlation for these 15 genes that I'm showing here. So this tells us that we can actually trust the spatial reconstruction data. Now we're looking at transcripts, and the reviewers also noted that it doesn't really mean that the protein that is encoded by the transcript is going to have the same localization pattern along the villus. So we checked for a few of them, and of course for some of them you can imagine that translational efficiency might be changing as we go along the villus, but in general, here you see, for example, a gene that the RNA is high in the crypt that goes down. Protein level, we see the same thing. So it's not that this is just, showing us RNA for a very stable protein, because you could imagine that if you have a very stable protein, you can just translate that at the bottom of the crypt, and then you don't need the RNA anymore, because as the cell migrates up, the protein is stable. But that's not the case. So the protein expression gradient actually nicely mimics what we're seeing on the RNA level. And down here you see NT5E, that's CD73, which is really in the epithelium expressed at the very tip of the villus. You see here there's a lot of expression in the stroma, so that it's completely not expressed in the epithelium. These are actually intraepithelial T cells that have a lot of that, but they're not in our single cell analysis. But here you see when we get to the very, very tip, there's tons of expression of CD73 or NT5E also on the protein level. Good. So again, we can trust these data. And now, what's the biology? What is happening in a spatially controlled manner? Excuse me. So first of all, we looked at different nutrient transporters. So here you see transporters for amino acids. They are in black, expressed mostly at the bottom, then transporters for carbohydrates in the middle, peptides a bit higher up, and then apolipoproteins that are used to then transport in the blood fats that are being absorbed. They are at the very tip of the villus. So different nutrients seem to be, at least we see that their transporters are subspecialized or are expressed at different zones, different heights within the villus. When we looked at the very tip of the villus, we saw that there's this new subpopulation that expresses different genes of the purine metabolism pathway. This is an important adenosine deaminase, and then here we have NT5E, which converts pretty much ATP into AMP, and we have SLUC28A2, which is an important purine transporter. So we also visualized this zone here by SM fish at the very tip. And since we know that ATP, cell-free ATP, is very immunostimulatory, and adenosine, which is coming out of this conversion, is very immunosuppressive, we think there's really an immune modulatory process going on here at the very tip of the villus that we're very interested in studying further. At the bottom of the villus, we see this zone here, it's called V1, which is just above the crypt. And we saw that there's a lot of secreted antimicrobial peptides that are expressed just above the crypt. Now, I told you the crypt is this zone, this protected niche of the intestinal stem cells. So we were wondering if this zone could act as kind of like a protective layer that prevents the microbiome to go into the crypt and to disturb the cells. So we looked at the REC3G expression that you see here. It's not a great image, but you see there's a lot of REC3G expression just above this crypt in a conventional mouse that has an intact microbiome. And then we took another mouse that is germ-free. And if it doesn't have any microbiome, this expression zone above the crypt doesn't exist. So we showed that this is really dependent, this zone is dependent, at least for this gene here, on the presence of the microbiome. And we speculate that this could be relevant for acting as a gatekeeper of the crypt itself. Now, a reviewer asked us if these different zones could actually come from specialized stem cells. So we hypothesized, since the cells are actually migrating upwards, that they must change as they migrate. But it could be that there is different stem cell populations that we haven't seen so far that would give rise to, the population that expresses the middle zone gene and so on. So to address that question, we took a mouse that has lineage tracing clones that were carefully titrated to always come from one stem cell. So here in white, we see a clone that comes from one stem cell, and here we see another one. And we took clones, and then we checked with fish and saw that this clone actually spans the tip zone in green and the middle zone in red. So we have within the same clone cells that express RNAs of the middle villus and cells that express RNAs of the tip of the villus. And here we do the same thing on the border between the bottom of the villus and the middle. So we show that there is some cells here in green within that one clone from one stem cell that express markers of the bottom of the villus and cells that express markers of the middle of the villus. And this shows that the cells change their expression profile as they migrate upwards, irrespective of the stem cells. So they come from the stem cell within one clone, but change as they migrate upwards. So the existing model was that you have a stem cell that is amplified as in a transient amplifying zone, and then it terminally differentiates into the different, tuft cells, goblet cells, enteroendocrine cells, or what we studied here, the enterocytes, and that's it. We, based on this data, actually think that the enterocytes then progressively transdifferentiate as they migrate through these different zones in the villus. And of course, the next question is, well, what is causing these gradients of gene expression within the villus epithelium? Are there any growth factors, any morphogens? Who is instructing that? And so here you see that the epithelium actually covers, it's just an outer layer, and inside we have this mesenchymal layer. And in the mesenchyme, we have a mixture of blood vessels and fibroblasts. So we then did the same thing as we were doing before for the epithelium and looked at the stroma. So we used laser capture microsection and isolated different parts of the stroma in a spatially defined manner, and then did bulk RNA sequencing. And here you see a few genes of interest. So this is the crypt stroma, and this is the tip stroma. And one gene that really caught our eye was LGR5. Now, LGR5 is a very famous intestinal stem cell marker. And because of that, because the intestinal stem cell is probably the best studied adult stem cell, we have a lot of mouse tools. So we have a knock-in mouse that expresses GFP under the control of the LTR5 promoter. And the result is what you see here. So in the crypt, within the epithelium, you have these beautiful LTR5 positive green cells. And we now also focused our interest on the stroma. And in the stroma here, you see these GFP positive areas at the very tip of mesenchymal cells, fibroblasts, that are positive for LTR5. Now, since there's a lot of mouse models, we were very fortunate that somebody also made a mouse that expresses diphtheria toxin, the diphtheria toxin receptor under the control of LTR5. So this mouse model basically in any non-LTR5 positive cell doesn't express anything, but in LTR5 positive cells, you have the diphtheria toxin receptors in the membrane. And when you then inject these mice with diphtheria toxin, this doesn't do anything to normal cells because they don't have a receptor. But if you are an LTR5 positive cell that expresses now this diphtheria toxin receptor, the toxin gets into the cell and actually kills the cell. So that's a very nice model to specifically ablate a cell type of interest. It's not a gene knockout, but we specifically kill the LTR5 positive cells. So when we were doing that in the villus tip, so here you see again the whole villus with the villus tip cells in green. When we do that, these villus tip fibroblasts are actually lost. And this correlates with the loss of the tip gene expression signature. So here you see EGFR, which is expressed at the very tip, these little white dots. And in green, you have here these fibroblasts that are LTR5 positive. And here you have another gene, adenosine deaminase in white, which is expressed at the tip. And here in green, again, these stromal cells. Now, we lose the stromal cells and the gene expression of the tip marker in the epithelium that is in proximity is lost for both of these genes. So that correlates. And then we saw that if we wait longer, so if we take a tip and we inject and wait for 3 weeks, then we have in the same mouse some tips where the LTR5 stromal cells don't come back. And here we don't have much white SM fish signal for ADA, but in some other villi within the same mouse, internal control, we have a regeneration of this LTR5 positive fibroblasts. And in these tips, we then beautifully see the gene expression of the epithelial cells that is coming back, thereby showing to us that this gene expression really depends on the presence of this LTR5 positive stromal fibroblasts that likely signal morphogens that induce this tip state in the epithelium. So what have we learned? In tissues, the gene expression pattern can depend on spatial coordinates, and the tissue can then utilize that by distributing distinct metabolic functions to different locations. And the spatial heterogeneity can actually be instructed by stromal cell populations. Of course, it's then still a chicken egg question. So what is telling the tip stromal cells that they are tip stromal cells and so on? But I think we at least have uncovered some layers of control. And so the epithelial part was published here, and this was just recently published, this tip signature. So with that, I'd like to thank Shelley Fitzkovich and all of the lab members at the Weizmann Institute that helped a lot for these projects that I talked about. I'd like to thank my lab members in Zurich. We are taking these projects further. We're also developing methods, especially in the subcellular space, to study gene expression, the more complete level, to identify functions of RNA localizations and determine mechanisms of how localization happens, not just by disrupting the cytoskeleton, but by looking at RNA binding protein interactions in a spatial manner. And I'd like to thank the funders and all of you for having listened to my talk. Thank you very much.
Thank you, Andreas. And then shall we start question and answer sessions then?
Excellent.
And if you guys have any questions, please feel free to unmute yourself and ask directly because we have time.
So Andreas, this is Rong. I I think you have the single cell sequencing data. So after you laser dissect, and then you can really see where the some single cells are from which layers. But I think people in the field also developed just a purely computational approach to look at the differentiation trajectory. I think your data is beautiful data, and this is real data. I think they were doing just a computational inference, but you have real data. But I'm just curious if you put your single cell data into the trajectory analysis, like for example, monocle or fade, and look at how those cells line up and teach you different differentiation stages, and then now you have real data to compare.
Yes, so let me just share my screen again. I actually have a slide. I just had to quickly pull it up. So this was, of course, a beautiful study that you referred to. Okay, here. Now let me go in full screen. Can you see this again?
Mm-hmm.
So the question, so this was Moor-Nitzan and the Nick Rajewski lab that were actually looking at if you just take the assumption that any gene expression gradient is usually going to be gradual, and then you have all of these different cells, and you can basically order. So here you have the different cells, and each one of them has information about all the genes. And so computationally, they just reordered all of them so that most of the genes are in continuous gradients. And they did that on our data, both from the liver and from the gut, and they showed that they can actually reconstruct without having any knowledge about landmark genes. So this is, of course, very beautiful. I would argue that our method is still of use because we can actually control if we capture the whole range of the tissue. Because if you look at, for example, published data sets, so here just to compare, so this is our original reconstruction in the x-axis, and they did our reconstruction computationally being agnostic to landmark genes. So they get really good overlap. But we also spent a lot of time on troubleshooting and identifying appropriate data sets. So based on landmark genes from laser capture, we know that we capture everything from the bottom to the tip of the crypt. So here you have this whole coordinate, and here you have numbers of cells. If you take a crypt-enriched data set, such as the one from Aviv Regev from the gut cell atlas, here you have mostly cells at the bottom, but you never get up to the villus tip. So if you only computationally rearrange, you don't really know if you cover the full extent of the tissue. And I think by doing laser capture, you can really define the extremes and then get the landmarks all the way up. But I was very impressed by the paper, of course, and I think it's really beautiful that you can do that without any prior knowledge of landmark genes.
But with your data, they can eventually verify that's done right.
I mean, of course, it also depends on gradual gene expression.
Mm-hmm.
As soon as you have we are looking at one cell type, and there it's gradual. But as soon as you have a tissue where you have two very different cell types next to each other, which even in my tissue you do, because you have a goblet cell or punnet cell that is very, very different in gene expression space from its neighbors. So there, this whole assumption that everything is based on gradual changes falls. And if you have different cell types next to each other, you're not going to be able to reconstruct without knowing the gradients, I would say.
Mm-hmm.
I got actually a quick question. So most of your models are mice, I guess, right? Do you have any human tissues that you have been studying?
For the RNA localization, we actually look at human tissues, and I think some genes are very nicely conserved in localization patterns, and some genes, interestingly, are very different. So we're very interested in that, but so far, of course, mouse tissue is more accessible and it's the RNA quality is usually higher, but we're comparing across different mouse tissues, but also across species. Yeah.
Interesting. And then the organoids are also mouse origin or human origin?
We have both. So mice are a little bit easier to create, and our RNA, so we have a big stock of RNA fish probes for mouse, and for humans we also have some, but they're not compatible. So,
sure.
But it's definitely also feasible for human organoids. They're a bit more tricky in terms of the growth factors and so on, but it's also feasible.
Yeah, I'm always interested about the translation of these results to humans, right? And in the sense that how translation really works in humans.
Yeah, so we're trying to, I mean, we can also use iPSCs to create fetal-like organoids from human lines, and we're trying to work out better methods for spatial sequencing by using APEX2 biotinylation to get the subcellular transcriptome, because laser capture of organoids is pretty tough to get enough material together, and it's really it's a noisy method. So we're trying to look at better subcellular profiling techniques, and I hope next time I can talk more about these.
Yeah, I don't know that much about the sort of the tissue histology here. What's the typical size, the dimension, length, and the width?
So the villus is about 500 microns. So I was cutting about 100 micron pieces. So that's much easier. The subcellular cells were about 20 microns high. So these bulk patches were about 5 to 10 cells of RNA that we got out. Yeah. So the histology is of course crucial, because if you have to have a very good orientation for everything. Otherwise laser capture doesn't work. You really have to see what you have to be able to isolate.
So those are kind of formalin fixed tissue. No.
If you use fixation, then you break the RNA, and then you have to do total RNA-seq, and then you have mostly ribosomal reads, ribosomal RNA. So this is unfixed, fresh, and it takes about 5 minutes. The staining is very quick. It's very quick staining. Then
kind of lighter the H and E.
Yeah, not H&E. H&E is not that, it's just Crescel violet is the staining.
Oh, okay.
And then you have about 30 minutes of cutting, and then the RNA is gone. So you have to work very quickly.
So I see Ruben wanna ask a question, right?
Yeah. Hi. I will just. Very nice talk, Andreas. I actually have a couple of questions, but I first want to go back to the first part. So I think in the first part you showed that there is this intracellularly, there is this transcript localization to the apical side and basal side. So I was just wondering if you go one step back and you actually look at the nucleus, where we know there's a lot of chromatin reorganization. Is there already, if you look at transcription, is that already organized in like basal and apical side?
It's a very good question. I haven't checked, but with SM fish, you sometimes see very bright transcription sites. And one could look, there's other techniques, but I think it would be interesting to see if there's, if this bias is started with preferential export to a specific site, or yeah.
And then I have a question about the second part. My question is, I think you don't really know that, because you said that at the end, is that what I'm wondering is, so is there actually crosstalk between the stromal cells and these enterocytes, so that if you would ablate the enterocytes, right, would you also lose the stromal cells? In the other way, basically.
I don't know, but since the enterocytes are constantly shed at the tip, but they're always repopulated from the bottom. So I think that would be a very hard experiment to do, because they're, every day they're replenished.
Right.
So I think it's mostly a one-way street of signaling, but yeah, I have to look deeper. I mean, we can look now with ligand receptor analyses of the single cell data to see if there is much going on in the other way. But so far, I'm not really aware of signals. Yeah.
And then can you speculate about what you think that actually drives the patterning of the stromal cells? Is that a signal from the blood, Is that maybe coming from the lumen of the intestine, the small intestines?
Yeah, I mean, the villus grows during embryonic development, and I haven't looked, but it would be interesting to see at what embryonic and developmental stage that you see these differences of the stromal cells. But of course, we know that there is hypoxia. So the higher up you go, the less oxygen is still reaching, and we see also some hypoxia responsive genes coming up there. That could be one way to regulate it. And that's also how we speculate that, you know, the design principles of the tissue, because the fat absorption doesn't need any active transporters. So we think it makes sense to do that at the tip where you have anyways less oxygen for metabolism and so on. But that's also just speculation. But that could be some feature that is spatially heterogeneous.
Great, thank you.
Thanks.
Yeah, I think this is already kind of beautiful system, but you probably want to really focus on it. But I think another system, if you are interested, that might give you sort of great insight to really compare. That's the hair follicle. I think the hair follicle and the stem cells right down there, once they differentiate, they push up towards the surface of the skin. Probably very similar biology there. And also human samples, that would be a lot easier to get than the gut tissues.
Yeah, that's true. That's a good idea. Yeah. And in terms of RNA localization, we're now also comparing to neurons, where the localization patterns are much more established and where there's a lot known about mechanisms to see if some of the RNA binding proteins that are relevant in neurons play a role in our system and so on. But we definitely look across borders, and we're interested in, I mean, even if we go along the gut from the small intestine, from the duodenum to the ileum, we see that some things completely change in these spatial patterns of RNA localization.
Yeah, while waiting, I can ask a question. So, you know, gut is important for microbiome research. Have you thought about looking at spatial organization effects of microbes on your system?
Yeah, I mean, I would love to know if the biofilm that is attached in spatial proximity to the epithelium changes as we go up and down on the villus. So I haven't had the time to look. I know that there's a lot of people that do spatial metagenomics now. It would be very interesting. I haven't had the time to look into that myself. But of course, if you have these absorption processes, they could also create different local microenvironments in terms of nutrient availability. So that could also be harnessed potentially by the microbiome.
And yeah, I could maybe ask one more question. So these weren't necessarily so highly multiplexed fish, right? Would you consider applying multiplex fish to see more co-regulation of genes?
Yeah, we are working on establishing a simple MER-fish, so without combinatorial readouts, but just doing some repetitive cycles to get about up to 50 genes at the moment, also due to collaborators here in Basel that have the microfluidics set up, the liquid handling, and we're looking forward to do that in the cells. But as I said, we also try to use proximity biotinylation of RNA with Apex for the subcellular realm to get better quality. Because some of the questions really need isoform data, for example, from the neuronal systems. You know that different isoforms can go to different areas within the cell. And if you have that isoform data, you know what is different, and then you can look for the functional regulation in the differences. This is not at all possible with the current RNA quality that we're getting from laser capture. So we really try different methods to go there.
Yeah, I see Suhi here from Redco, and so I think that they probably can provide you complementary technology. Although I believe Suhi gonna be speaking in Series 3 as well.
Oh,
Yeah,
actually, someone from our company, Sam and Versa, will be speaking on behalf of the company. But if you'd like to, we would love to talk to you if you have time.
yeah, it'd be excellent. Actually, I was in touch with some people from the company already, and I think it would be great to look into that further.
Fantastic.
Great, thank you very much. Really appreciate it, and looking forward to the upcoming talks.
Yeah, thank you, Andrea. This is a terrific talk.
Thank you very much.
Yeah. So I think next week, I think, yeah, keep in mind we're gonna start at 2 o'clock again, and also very likely we're gonna finalize our flyer and circulate. And we may change the Zoom link or Zoom link password. So make sure you use the new Zoom link information from the flyer.
Thank you very much.
Thank you. Thank you, Andreas. You have a good night.
Thank you so much.
Thanks.
Sure. Have a nice weekend.